Hello everybody. In this video we are going to study two types of bryophytes, type study of bryophytes. The first one is Anthoceros and the other one is Funaria. So to begin with Anthoceros, we should know the systematic classification to which category this Anthoceros belong to. So to start with, division Bryophyta, class Anthoceropsida, order Anthocerotales, family Anthocerotaceae, genus Anthoceros, species Anthoceros mangularius, Anthoceros gemulosus. Okay? So these two species have been given as examples. Now, talking about its distribution, it is present worldwide. What type of temperature, what zones are they comfortable to live in? It is the tropic and the temperate zones wherein you can find these species. Across the world there are about 208 species, whereas in India we have only 32 species. Now, talking about the plant body, so the gametophyte, the particular plant, that is the Anthoceros, consists of two phases. First one is gametophytic and the other one is sporophytic. So the gametophytic consists of the haploid chromosomes and the sporophytic is diploid in number. Now, talking about the gametophyte, these Anthoceros are also dorsoventrally flattened as that of Mercantia, which you have studied in the previous video. So now in this gametophyte you will find they are just thalloid in nature. You can't be distinguishing them into dichotomous branches as that of the Mercantia, okay? Nor that they have sporophyte, that is a slender, thin, tubular structure arises from the thallus, okay? So this is the sporophyte, okay? They are thin in nature, okay? They are thin, slender, sticks like arising from the thallus. Now, this slender sticks give the name as hornworts for this Anthoceros. Now, the thallus, the dorsal surface of the thallus is velvety in nature, okay? They are dark green in color and also that they are made up of 2 to 3 celled thickness layer. Whereas we talk about the ventral surface, the surface below, okay, the ventral surface consists of rhizoids, okay. The rhizoids are simple rhizoids without scales with them, okay. The dorsal surface, they do not consist of scales or the tubercular rhizoids, it's just the simple rhizoids. So you can see the thallus, they are just spread apart, thalloid, that is dorsoventrally flattened, and they do not show dichotomous branching here, nor they show the midribia. So they have a special structure, the sporophytes, which is thin, slender, stick-like structure arising from the thalli, and hence we call it as hornworts. The Anthoceros are also called as hornworts. Now, moving on to anatomy. Anatomy of the thallus. When you take a cross section of the thallus, you can find that they consist of a single celled epidermis, okay. They have upper and lower single celled epidermis, and later on you can find they are multicellular layered cortex region which consists of cells which have a very dominant nuclei, okay. Each cell consists of a dominant nuclei, a chloroplast and a single pyrenoid, okay. The entire thallus consists of isodimetric cells, okay. These cells are similar to one another. They are the simple parenchymatous cells wherein they have a well developed nuclei at the center and they consist of a single pyrenoid and chloroplast. So that is present in all the cells. One special feature you find among them is a large cavity, Okay, you find an empty large cavity which appears to be greenish blue in color. Why is it so? These allow the nostoc or anabina. Okay, the blue-green alga, nostoc and anabina is found growing here. That's the reason they gave a bluish green color, and they have mucilage present in it. Okay, so there is a minute pore or openings on the thalli which is called the mucilage pore, through which these can move out. Okay, so that is the thallus structure, anatomy of the thallus structure of Anthoceros. Okay, so they harbor blue-green algae, that is nostoc, anabina. They consist of a single epidermal layer, upper and lower epidermal layer, and they have plenty of cells which is consisting of a nucleus, pyronoid, and then chloroplast. Next, moving on to reproduction, it is the oogamous type. Okay, a specialized oogamous type. You can see the sexual reproduction occurring wherein the male reproductive structure is called as antheridium and the female reproductive structure is archegonium. Now, talking about antheridium, they are again present inside the thallus. Okay, inside the thallus, in a cavity shaped, you can find the antheridium to be present. So they have a roof, the cells of the roof, okay, and they have thallus, that is the thalloid cells present. There is the floor on which you can find a multicellular stalk being present. Okay, so they have a multicellular stalk. They consist of a thin single jacket layer. Okay, so they have a single jacket layer. Sterile cells, these are the sterile cells which is present, and inside you have plenty of sporogenous mother cells. Okay, so I'm just drawing like this, but they are not like this. Okay, so you'll find plenty of sporogenous mother cells. You can find each cells to be present which are diploid in nature. Okay, diploid to n number. Okay, so this diploid cells will undergo meiosis. Okay, when they undergo meiosis, these sporogenous mother tissues will give rise to androcytes, and this androcytes will give rise to androzooids. Okay, so androzooids are the male reproductive structure. Each cell can be formed into a single anthrozooid, which is spindle curled structure which consists of two flagella. Okay, so that is the structure of anthrozooid. Now this antheridium is present inside the thalli. Okay, so they are present inside the thalli. There is a cavity which has been present, antheridial chamber. We call this as antheridial chamber. Okay, so this is the roof. This roof cells will disintegrate and this anthrazooid can swim out. Now, this antheridial chamber is present inside. Now, same way you will find the archegonium to be present. The archegonium is the female reproductive structure which is a flask shaped structure. Okay, you will find them to have a swollen base, okay, a thin slender neck. This is again present inside the thalli, okay, inside the thalli. Hence you can find there is neck cells, that is the thallus cells, okay. Then you will find the venter cells which is present around the venter. You will find the cells of the thalli to be present, okay. So you will find them to be present like this. Now they consist of four neck cells and 2 rows, okay, that is these are neck cells. The cell adjacent to the neck region on the thalli. Then they consist of a special cell, that is the egg cell, which is big in size with all the reserve food material, and they do consist of a special cell, that is venter canal cell. Okay, so the swollen tip is called as the venter, and inside this venter you will find a venter canal cell and an egg to be present. Now above this venter canal cell you will find neck canal cells to be present. Okay, so there can be 4 to 6 neck canal cells present in the neck region of archegonium. And again you will find at the tip there is 2 special cells, that is the cells which is closing the neck of the venter, closing the neck of the archegonium. So what happens is you have 4 to 6 neck canal cells, 1 venter canal cell and 1 matured egg which is present in the venter region, that is the swollen base. Now this bryophytes, as we said in the general characters, they require water for fertilization. So in the rainy seasons this anthrozoid will swim out of the antheridial chamber and they are going to swim across and reach the archegonium. So at the time it reaches the archegonium, this venter canal cell and the neck canal cells starts to disintegrate. Once this egg is matured, the venter canal cells and the neck canal cells, they start to disintegrate. Okay, they completely disintegrate, disorient itself, and there will be a sweet liquid which is produced, and that is some chemicals which is secreted there. These chemicals will attract the anthrozoids. Okay, when this chemical attracts the anthrozoids, they come reach the neck and then they swim across the neck canal cells and then they come in and fuse with the egg. Okay, so once they fuse, there is a diploid zygote which has been produced, and that zygote, that is the fertilization occurs, and this zygote will divide itself to form the sporophyte. Now, talking about sporophyte and anthoceros, it is a bit different from that which we studied in the mercantia. Only the sporophyte varies with the anthoceros, mercantia as well as funaria. So what happens in the sporophyte? This zygote starts dividing, multiple divisions. Okay, once they divide, they will form into a slender, long sporophyte. Okay, okay. So now they are still in attachment with the gametophyte. So the first contact of the sporophyte and the gametophyte is the foot. Okay, so foot is a bulbous structure. Okay, the foot is a bulbous structure which is made up of meristematic tissue, and they are the ones which is allowing the anchorage. Okay, they are the ones which holds on the sporophyte on the gametophyte, and they will supply the nutrients, water, etc. from the gametophyte to sporophyte. And they have a protective structure called the involucre. Okay, so involucre is present which protects this sporophyte. So after that you will get an intermediate zone. Okay, so intermediate zone is present, from wherein you will find a special structure here, that is the columella. Okay, so you will find the columella present here. So this columella consists of 16 vertical cells. Okay, so they have 16 vertical cells across. Okay, and then talking about this the columella, that is the capsule. Okay, you will find in the capsule they are consist of 3 regions. The first one is columella, second one is sporogenous, and the third one is capsule wall. Now talking about columella, as I said, they have 16 cells which is vertically placed. Okay, and then you have the sporogenous tissue starting off. This sporogenous tissue is 2 to 3 layers. So you will find sporogenous tissues, that is sporogenous mother cells, to be present adjacent to the columella, which is the one which is diploid in nature. Okay, so you will find 3 layers there. Apart from that, you will find the capsule wall, which consists of epidermis layer. Okay, so they have epidermal layer, and then you will find that they consist of 4 to 6 celled thick cells. Okay, that is, you have 4 to 6 layers of cells that is present adjacent to the sporogenous tissue and the epidermis. Now, this epidermis is interrupted by stomata. Okay, they do have stomata at the center. Okay, and they are interrupting the continuity. Then you will find that these cells which is present in the capsule wall are chlorenchymatous in nature. When we say they are chlorenchymatous in nature, they take up photosynthesis. So, the sporophyte of Anthoceros are partially autotrophic and partially dependent on the gametophyte. Okay. Now, what happens to the sporogenous mother tissue? This sporogenous mother tissue will undergo meiosis. Okay. They will undergo meiosis and there are spores being produced. Okay. So, there is this layer wherein spores are produced. How are these spores produced? They are homosporous, similar, but they are formed in tetrads. Tetrads, one region you will have 3 spores, okay, and the other one will be on this, okay, 3 spore, another one on this. So, there will be completely 4 spores being produced together, aggregated together. At maturity, okay, we say spore tetrads are being formed, okay, spore tetrads are formed, and at maturity each spore segregates itself. It separates from it and a single spore will be dehisced. Okay. So, dehiscence of sporophyte, this entire structure, that is, we say they are hornworts because they have a slender long stick like structure of sporophyte. So, once they are matured, they dry and dehisce in longitudinal, that is vertical straight lines, they dehisce and open up. So, a particular tubular structure will open up like broken stick which opens up. Okay. So, that is how they dehisce and the spores are liberated from this sporophyte. Okay. With that we finish Anthoceros. Next we will move on to Funaria. So, when you begin with the type study of Funaria, this is how the classification and how it is placed systematically. The first one is division Bryophyta, second class Bryopsida, order Funariales, family Funaraceae, genus Funaria, and species we are talking about Funaria hygrometrica. Okay. Now, which is the conducive climatic condition? Again, it belongs to the tropical and temperate zones, same for all the bryophytes. Now, talking about distribution of the species worldwide, it is about 117 species, and in India you can see only 15 species of Funaria. Now, moving on to the plant body. So, for the first time we know that evolution occurs wherein there is this minute changes which is leading for a new species. So, when you come across all the groups species wise, you will find there is a evolutionary trait, that is particular changes which is paving way for another set of groups. Okay. So, here you can see the first move. Okay. So, we read about or we studied that the Mercantia, the Anthoceros are all thalloid-like structures. They are made up of thallus, that is dorso-ventrally flattened body. Okay, but whereas you look around, you can see some of the angiosperms which consist of leaf, stem, branches, flowers, fruits, etc. So here for the first time you will find a plant body which consists of leaf. Okay, this is the main axis, we don't call it a stem. Main axis which consists of rhizoids, root-like structures, and then you have leaf which are spirally arranged on the main axis, or you can even call them as stem. Okay, so this is the plant body. You have leaf which is spirally arranged across the main axis, and then you have the sporophyte which is almost like a drooping fruit. Okay, so this is the sporophyte. So this happens to be the gametophyte. You have rhizoids, stem, leaf, and then the capsule which belongs to the sporophyte. Now, so first you can say they have a protonema, a flat horizontal structure under the ground, that is below the soil surface, which give rise to an erect branches. This erect plant body consists of the two types of gametophyte. Okay, one is the female gametophyte and the other one is the male gametophyte. How are we distinguishing? It's just because the sporophyte is present. The sporophyte will be present on the female gametophyte rather than the male gametophyte. Now, what is the length, height of the plant? It is about 1 to 3 centimeter tall. Now, moving into the anatomy, that is the stem, the main axis is also called a stem, wherein when you take a cross section, you will find it to have an epidermal layer, a single epidermal layer. And then you will find at the center thick walled cells, okay, which is helping in conduction, and then the cortical region wherein you will find thin walled cells which is present. Okay, they are parenchymatous in nature. Okay, so this is how you will find the cross section of the stem. Okay, so for the first time you will see the stem resembles the stem of the angiosperms. They do have epidermis and they have a special cells which is present in the center which is taking up conductivity. Okay, so that is about the stem. Now, moving on to the anatomy of leaf. So the leaf again shows a midrib and leaf wings. Okay, leaf wings. So at the midrib you will find this conducting tissue present. Okay, and then you will find simple chlorenchymatous cells, that is parenchymatous cells, which consist of chloroplast. Okay, chlorophyll pigments are present here. Okay, so that's how you will find the leaf to be present. So at the center it is swollen, that is the midrib region, and then you have the wings of the leaf which consist of parenchymatous cells which has chlorophylls that is present at the upper and the lower surface of the leaf, same like that of the angiosperms. Now, moving on to reproduction, again it is oogamous type, sexual reproduction wherein there are two organs, that is antheridia and archegonium. So there is one special feature in the Funaria, they are called as protrandrous. Okay, protrandrous means the male reproductive structure matures first. Okay, so what happens, the same plant consists of both. Okay, they can be monoecious or dioecious in nature. When they have two sex organs on the same plant, still they do not fertilize, they go for cross fertilization. Another plant, they are going to fertilize another plant. Okay, now talking about antheridia, there is a few difference here. So the terminal structure, the leaf terminals, that is the stem at the terminal consists of plenty of leaf like a bunch at the tip. From there the male and the female reproductive organs is present. Now, so you can find there are leaf-like structure which is protective in nature, and they are called as perigonial leaf in the case of male reproductives, that is antheridium. And you will find at there is the antheridia consisting of a multicellular stalk. Okay, so they say it is also acropetal in arrangement. Okay, so I'll draw one big one so that you can understand better. They are intermixed with sterile paraphysis, sterile air paraphysis, which is said to be protective in nature. Okay, so these are paraphysis. Okay, they are protective in nature. Now, talking about the antheridia, you will find that it consists of a single sterile jacket layer. Okay, a single sterile jacket layer and plenty of anthrocytes. Anthrocytes are nothing but the spore mother cells. That is the antheridial cells which is diploid in nature, and they are going to undergo meiosis. Okay, so they are going to undergo meiosis to form the anthrozoids. You will find plenty of sporogenous tissue. Okay, so it's better to draw like this. Plenty of sporogenous tissue, and each cell in the tissue will undergo meiosis, and that is the reductional division, and there is formation of anthrozoid, which is again a spindle shaped with two flagella. Now, moving on to archegonium, again they are present on the tip of the plant, that is wherein plenty of cluster of leaves are present. So you will find them to have protective leaves around them. So they are called as pericaceal leaves. Okay, so they are the pericaceal leaves. Apart from that, at the center you will find a small stalk, okay, with a swollen base and a slender neck. Okay, so this is the archegonium. Okay, so they do consist of sterile hairs in between, which is again protective in nature. Okay, herein you can see that they have a one big matured egg at the center, which is accompanied with a venter canal cells. Here you have 10 neck canal cells present. Okay, so there are 10 neck canal cells which is present here, which is gonna help in the production of the secretion. Okay, then so in the case of fertilization, as I said earlier, they have protrandrus. What happens? The male reproductive structure matures first, and then the archegonium, female reproductive structure, matures. So when they are maturing later, that's the reason cross fertilization occurs. Now, what happens is, as in all the bryophytes, these cells starts to disintegrate or disorient themselves. Okay, so they disintegrate themselves, and there is a sweet liquid which is produced, and these anthrozoids swim and reach the archegonial tip. Okay, so it is the heavy rains which is supporting it, and then these anthrozoids swims across the neck and then reaches the egg. Once they reach egg, they're going to fertilize, and there is the production of zygote present there. So that's the reason we say sporophytes are only produced on the female plant, whereas the male plant cannot produce the sporophyte. Why is it so? Because the site of fertilization, the site of product of fertilization, everything is in the archegonium. So this funaria is also called as mosses, okay, wherein you will find after the zygote being produced, they are going to form into sporophyte. Again, you will find in the case of funaria, there is a difference in sporophyte. So how is the sporophyte there? They do consist of three common features of all the sporophytes and bryophytes, that is the foot, seta and the capsule. The foot is helping them for the anchorage. Seta is the connecting link between the foot and the capsule, wherein the seta will be supplying the nutrient etc. got from the foot to the capsule. Now, talking about the capsule, that is the sporophyte, you will find it to be present in a bulbous structure. Okay, so they are bulbous in nature. So the basal region is the place wherein you will find meristematic cells and the parenchymatous cells present. So this is the apophysis region. Okay, so this is the apophysis region wherein they are green in color. So since they are green in color, they do consist of chloroplast, chlorophyll, which is taking up photosynthesis. And then you will find that the middle region is called the theca. Okay, so this is the theca region, and the tip region is called as operculum. Okay, now talking about the theca region, you will find columella present here. So how is the columella present? In a cone shaped. Okay, you will find the columella being present in a cone shaped. So they are the ones which consist of plenty of cells, and they are the ones which is giving a support system for this capsule. So apart from this columella, you will find that they are having sporogenous tissue. So they have a single layers of cells next to this columella, and then you will have sporogenous tissue present. Okay, so they have sporogenous tissue, and afterwards there is again 3 layers of cells being present after the sporogenous tissue. Okay, I am just converting this because of the space. Now after that you will find a slender, long vertical cells being produced. Okay, so these cells are called as trabeculae. Okay, so these cells are in contact with another layers of cells present here, which is the hypodermis. There is around 3 to 4 layers of hypodermis, and after hypodermis you will have epidermis present, that is the outer region. Now trabeculae are the thin, long vertical cells which is present, and there is space here in between. So these are called as air cavity. Okay, so this is the theca region, which resembles a urn shape, a pot like, okay, urn shaped structure which consists of air cavity, sporogenous tissue, columella in that region. So again, this sporogenous tissue will undergo meiosis, and there is production of spores here. The other thing is, there are certain sterile cells between the sporogenous tissue. These sterile cells will form into pseudoelaters. Okay, they are the pseudoelaters which is being produced along with them. Now, Talking about the operculum, that is the tip of the capsule, this operculum is made up of 3 layered cell wall. Okay, 3 layered cells are being produced for this operculum. Inner region, inner to this layer, you will find a special layer called as peristome. So peristome are curved cells which is resembling teeth-like structure. Okay. So how many curved cells are there? They are about 16 curved cells in 2 layers. Okay, there will be 16 like this, first layer which is teeth-like curved cells. Again, next to it you will find another layer which consists of these curved cells. So what is the special feature? They are hygroscopic in nature. What do you mean by hygroscopic? They absorb water and release. So at the maturity, this operculum will fall off. Okay, they just fall off because of the action of peristome. The teeth will open up and close, and once they open up, all the spores which is in the sporogenous layer inside the capsule will be pushed out. Okay, so that is about the sporophyte of the Funaria. So in the last, I would like to do 2 more plant pathology, 2 plant diseases. So we had learned about these diseases in the previous video. Okay, so a continuation, a small patch to be done. So we will be talking about downy mildew, disease of bajra, and sandal spike disease. Now, when we talk about disease, it's better to know why are we doing it. So as the previous class which I have told you all, we need to know the diseases, the causal organism, what are the symptoms and how can we control it. So since the plant is the only producer across the world, we have to conserve and protect them. Okay, and due to increase in population, the amount of yield is also being counted with the quality, not just the quantity, the quality is also important. So in that note, we should be knowing that how are these plants being attacked by pathogen? If so, what could be done to prevent them and to get the product? So to know that a particular plant is being attacked by a pathogen, we have to take up the diagnostic way. Okay. So to begin with, we will be starting with the downy mildew disease of bajra. So which is the causal organism? Plasmopara verticola is the fungi which is causing this particular disease. So when we analyze the symptoms, we can be aware of that particular plant being attacked by this fungi. So once we know the symptoms, it is very important or very clear what measures could be taken to prevent it. So the first symptom is you will find a yellowish white patches on the upper surface of the leaf. So if the bajra leaf is there, on the upper surface you will find patches, yellow and white patches which is irregular on the upper surface of the leaf. Whereas on the lower surface you will find there is some cottony outgrowth. Okay, so cottony outgrowth which is white or grayish in nature. So this cottony outgrowth is nothing but the mycelium of the fungi which is growing out through the stomata or piercing out through the epidermal cells. Okay. So this is the first symptoms which appears on the bajra. So once the symptoms is appear, we are now getting confirmed that bajra is being attacked by the fungi. So when the infection becomes severe, you can see that the leaves turns crisp in nature. Okay. So the complete leaf will turn brown in color, that is the yellowish patch will darken up and turn brown in color, and then they become crisp in nature and eventually they fall off. Even though there is adequate amount of water being supplied to the plant, the leaf turns crisp. That is the time when we say the infection is severe. and this severeness should be an indication to removal of that plant and destroying it. Now, we will know some of the preventive measures. The first one is pruning. Pruning is nothing but removal of the affected leaf. Now, this fungal might be attacked only to 1 or 2 plant in the entire field. Then you can see only in the leaf region, you can see these patches, then you can just remove off the leaf region. And the other thing is the ground should be kept clean. In case the leaf has been affected, infested, they are fallen on the ground, the wind blows and that infested leaf will go and stick onto another plant. So, there is all chances that an healthy plant can be infested by the infected leaf. So, the ground should be kept clean, especially in the autumn, especially in the spring seasons, okay. And then we need to remove the weeds. So, the fungal can be spread when the weeds are removed off, there is spaces being created, okay. So, this also allows aeration and the reduction of the growth of fungal. Then it is better to water in the morning. So, when you are watering in the morning, it is going to take up maximum benefit. Then the last thing is to spray the foliage and fruit with copper spray. Okay. So, when I said the symptoms, it is merely or only seen on the leaf. So, we are going to spray the fruits and the leaf with the copper spray so that we prevent the fungal infection, proliferation and also damaging the plant. Now, moving on to the next disease, that is the sandal spike disease, which is prevalent or seen in India. So, it is caused by MLO, that is mycoplasma like organism. We are not definite what it is, okay. So, this mycoplasma like organism are the reason of causing sandal spike disease. So, the measurement is merely 60 to 750 nanomicron, but that is they are very small in nature and they are ellipsoid in structure, ellipsoid, okay. You will find them to be present like this, okay. Very small organism and it is causing sandal spike, which is a serious disease. Now, talking about the symptoms is the roset spike. Roset spike is the very evident and the most common symptoms of sandal spike. So, when you talk about spike, it is nothing but the leaf which has been converted into spike like structure. So, we know that the lamina, that is the surface area of the leaf, is broad enough, but when they are being affected by MLOs, the leaf, the dimension, the breadth of the leaf will be reduced, okay. So, narrowing of the leaf is occurred. So, what happens is the leaf becomes narrow, spikes like, okay, and the internode is also reduced. So, the internode region will reduce in size, okay. The internodal region becomes smaller and then the plant shows drooping nature, okay. The entire plant starts to droop down. So, internodal region will be swollen, then smaller means there is plenty of leaf coming closer. So, that we say roset. Roset means arrangement in whorls, okay. So, there are spikes which has been arranged in whorls, that is the spike in the sense the leaf, hence we call it as roset spike. That is the primary symptom. When the disease is infesting, proliferating and affecting in maximum, when it advances, the leaf will turn into reddish yellow color, okay. So, when the leaf turns into reddish yellow color, then the disease is very severe. Later on, the entire plant dries and dies off, okay. So, that is about the symptoms. Now, there is a special person here, special insects that is Monia alimaculata and then Nephrotitis virescense. Okay. So these two are the vectors, that is the carrier of mycoplasma like organisms. So they said that this is a Chinese leaf hoppers and this is the grasshoppers. So both are the ones which is the plant oriented insects, that is they are leaf hoppers, grasshoppers, that is they take in the mycoplasma and they can jump along the sandalwood field wherein the crop can be infested with mycoplasma. So these are the vectors. So we are supposed to keep these two at bay so that the plant can be saved. So the other thing is control measures is not that evident. There is no much research work done here. So what happens is they have come across only one alternative, that is to grow Mysore gum tree. Okay. So they say that Mysore gum tree can be grown from 10 to 20 meters apart from the sandalwood trees, where the sandalwood trees are there. From that field 10 to 20 meters apart, when you grow this Mysore gum tree, it is known to reduce the MLOs infesting the sandalwood. Okay. So that is the only control measure. Maybe they are keeping these vectors far off from the sandalwood. Okay. So that is about the two diseases wherein we know which organism is causing it. When we know the organisms, we are capable of coming up with some of the chemicals to kill it and to preserve the plant and have the yield out of it. And then you know the symptoms wherein they are the indicators which is for the first time going to show that this particular plant has been infested. When these indicators go on, we are capable of identifying it and controlling it by few controlling measures, and then we are getting the product. Okay. So with that we finish the plant pathology. Thank you.